Mm, Okay, session. Today is session 2 of Western Blot e-learning live webinar. I'm Kylin, the regional field application scientist of Azure Systems. Today we will look at a step-by-step guide, and I'll walk you through the Western blotting protocol, covering both chemiluminescence westerns and also fluorescence westerns. These are the agendas for today's session. Firstly, we will walk through the chemiluminescence Western blot protocol, look at what are the troubleshooting tips for Chemi Westerns, take a quick glance on Fluorescence Western blotting protocol, and finally, how you can improve your Western blot quantitation power with total protein normalization imaging. For some of you who are joining this session today, who are new to the session today, first, let me introduce Azure Systems to you. Azure System is founded based on innovation. It's co-founded by Arnold Shifji and Dr. Diping Chair. Arnold is our CEO, a venture investor, while Dr. Diping Chair is our executive VP of R&D, where he led the whole R&D department of Azure System of a series of our protein imaging systems and CLO real-time PCR system and so on. We founded in 2013, based in Dublin, California, in the United States, very near the Silicon Valley. There, we designed, developed, and manufactured all our products in San Francisco Bay Area. The core member of the Azure systems, including Dr. Deeping, are coming from Alpha Innotech, a brand that we're quite familiar of if you're doing chemiluminescence, which then acquired by Cell Biosciences and become Protein Simple in 2011. So back then, Protein Simples wanted to diversify the portfolio, so the less resources went to the development of Western blotting images. So then Dr. Deeping considered to found a new company, hence the birth of Azure Systems in 2013, where we developed the best imaging system that focused solely on such equipment and associated reagents. We Azure Systems aim to give you raw data that you can rely on using a simple workflow that you can trust. These are the few key instruments that we can provide to you for your research need. We have Sapphire Biomolecular Imager, Azure Imaging System, and Azure CLO Real-Time PCR Systems. All right, before I start the session officially, let us recap what have we talked about last session, which is in session 1. Firstly, we talk about the principle of Western blot, the choices and consideration of what kind of gel, membrane, transfer system, blocking buffer, wash buffer, and also the choices of picking primary and secondary antibodies. Next, there are two major detection methods right now, which are chemiluminescence and fluorescence methods. We have been discussing about the advantages and drawbacks of each of their own, time and cost taken, quantitation power, and which is right for your experiment. We also talk about how to image Western blot, about in terms of film limitation, dynamic range, differences in CCD camera, and also excitation light sources for instance, Western blot, the comparison between RGB and near infrared signals as well. And finally, we touch a little bit about Western blot normalization, where we discuss about the advantages and limitation of housekeeping protein and total protein normalization. I hope it gives you a general idea about the flow of the whole e-learning webinars. So these are the session 1. Let us move on to session 2. This is a simplified flow chart of Western blotting workflow. As you can see, these are quite simple. It starts off with electrophoresis, transfer, blocking, primary antibody, incubation. There we separate into two different types of detection system, chemiluminescence and also fluorescence detection methods. And finally, analyze the data. Before I go into in-depth walk through about chemiluminescence Western. Firstly, we have to know, or you have to know, which method is right for your experiment. This table listed the details or the specific requirement or need or your if you want to use chemiluminescence. It would be best to use chemiluminescence if you just want to detect a single protein. This and you just want to see whether the protein is present or not and is purified or not. But it will be best to use Fluorescence Western if you want to detect multiple proteins simultaneously, especially for those or two or more proteins that are sitting at the same location and cannot be separated by gel electrophoresis. And Fluorescence Western also gives you more or better quantitation power in terms of counting the relative expression between two or more groups. Right. Let us walk through the chemiluminescence Western blot protocol. Here I'll be using azure radiance, which is an ECL substrate as an example. Since 1988, enhanced chemiluminescence or ECL has become one of the most common detection method in Western blotting. In this method, the secondary antibody is conjugated to the enzyme horseradish peroxidase or HRP. So once bound to the membrane via the primary antibody and also that recognizing the epitopes of the protein that transferred on the membrane, when we incubate the blot with ECL substrate, which normally contains luminol, the secondary antibody, which have the HRB, will react with the substrate, where with this chemical reaction, where the luminol in the presence of HRP and also hydroxyperoxidase, and it will emit light. This light emitting in terms of chemiluminescence light, this is what we're talking about when we talk about chemiluminescence Western blot. So the theory is all boils down to the chemical or enzymatic reaction between HRP and also the luminol substrate plus hydroxyperoxidase. This chemiluminescence signal can be detected by two ways, either X-ray film or by CCD camera. Right. So on the left, we have a list of materials that you have to use or you need to use in chemiluminescence Western. Firstly, of course, you need to have your primary antibodies of your protein of interest. Second, secondary antibodies conjugated with a HRP corresponding to your primary antibody. Of course, when you want to do the gel electrophoresis and transferring steps, you need to have these electrophoresis apparatus, power supply, gels, buffers for standard Lamley the SDS page, and also electroblotting apparatus and transfer buffer as well. When we talk about blot or Western blot, the blot here means the protein transferred to a membrane. Here we have two general choices, nitrocellulose or PVDF membrane. In this case, we normally cut it into the size of the gel after the electrophoresis to fit it nicely and make it into a transfer sandwich. Blocking buffer to block off excessive unnecessary signals. Wash buffer, either PBS or TBST. T here stands for twin 20. Methanol to activate PVDF membrane if you're using PVDF membrane. Forceps with flat smooth tips. Best is plastic forceps. Incubation and washing trays with smooth interiors to avoid scratches. Rotary or rocking platform for gentle agitation, and finally CCD-based detection system or X-ray film. These are the list of materials that you need to use when you're performing chemiluminescence Western. And on the right, this is a very simplified workflow of what we would do in this Western blotting protocol. I'll go into details in the next slide. So this is a detailed protocol. Firstly, we'll load the sample into the gel and run electrophoresis. So this step what we call as protein separation steps. We separate the protein samples by this either polyacrylamide gel electrophoresis using a mini-sized gel, for example, 8 to 10 cm, 8 times 10 cm. While running the gel, we should prepare the PVDM membrane. In this case, we're using PVDM membrane. You can also use nitrocellulose membrane as well. In terms of PVDM membrane, pre-wet the PVDM membrane in 100% of methanol for 1 minute until the membrane is completely wet. Then transfer this wet membrane from the methanol tray to a tray filled with purified water. You can either use millicube quality or distilled water in this case and incubate the pre-wet membrane on this tray for at least 5 minutes on a rocker with gentle agitation. Make sure that the membrane is fully immersed in the water and does not float on the water surface. Then transfer the membrane to a tray filled with transfer buffer and incubate it for 5 to 10 minutes. At this time, your gel electrophoresis should be almost complete. When it's completed, where then you should assemble the membranes, then together with the gel and filter papers, making it is what we call as transfer sandwich. It's a sandwich containing multiple layers as required for transfer apparatus. There are two transfer systems here that you can use. First is wet transfer, which indicated by this figure, and secondly is semi-dry transfer. For Western blot, we recommend to use wet transfer. You can transfer it for overnight at 15 volt, or you can run it at a higher voltage to shorten the amount of time, such as 1 to 2 hours at 70 volt in ice pack to prevent any deformation. Just a quick note about this. We Asus Biosystem we provide Azu Aqua gel tank, transfer gel tank, which it comes with two ice packs that is compatible and nicely fit inside the transfer gel tank, which is very good if you want to run it at a higher voltage. Right. After you transfer the protein from the gel to the membrane with the transfer system, here we use wet transfer. Then next is a membrane blocking step. After you take then you should disassemble the transfer sandwich. Take out the membrane. Please keep in mind that the membrane should be wet at all times, shouldn't be dry. Put it in a tray filled with water, purified water for 5 minutes just to wash off the transfer buffer. And then after that, transfer the blot to a tray filled with blocking buffer. Here we use non-fat dry milk blocking buffer with gentle agitation for 1 hour at room temperature. So in terms to give you the best or adequate blocking, you must at least use 0.2 to 0.5 ml of blocking buffer per centimeter squared of the blot to give you sufficient amount of blocking. Then at that time, prepare your primary antibody mix in the blocking buffer that you use. In this case, we're using the non-fat dry milk blocking buffer. First, we're preparing the primary antibody or diluting it in this skim milk blocking buffer. Well, preparing the solution, gently mix the solution by inverting the tubes several times. Do not vortex it. The optimal primary antibody dilution must be determined empirically. In CCD imaging, we recommend primary antibody dilution from 1 to 1000 in 1000 to 1 to 10,000 dilution. A good initial dilution will be 1 in 5,000. However, if you are going to detect the signal with X-ray film, use 2 to 5 times lesser primary antibody than for using in the CCD imaging. For example, I have been using 1 in 1000 dilution of primary antibody in CCD detection, which is optimal for my Western blotting protocol. If I want to change to X-ray film detection, the primary antibody dilution should be 1 in 5,000. I'll start off with 1 in 5,000 and see the signals are okay or not. Right. Next, after the blocking step, so this is a blocking step, we remove the solution from the tray, or you can just transfer the membrane to a tray filled with these pre-prepared primary antibody mix. Put on the pour it on the membrane and incubate it for 1 hour at room temperature with gentle agitation. In terms of primary incubation and secondary incubation, there are a lot of, I would say there are different types of incubation time and also taken, and also the temperatures taken. Some might be doing it in 4 degree for overnight, which is specifically for 16 hours. You can also do that if you want to have more specific bindings. But in this case, we going for a fast and quick Western blotting protocol. Right. Antibody can be added into a dish, placed on a shaker, or you can also use a smaller volume, such as 5 to 10 mils. Use it in plastic bags, seal it with a blot with the what is that called? Sealer, the heat sealer, and place it on the rotary platform. If you're using lesser volume, this case you will save a lot of primary antibody in terms of cost efficiency. Right. Next step is rinsing step, where we're supposed to wash off the excessive primary antibody from the blot. Upon the completion of primary antibody incubation, transfer the membrane to a tray filled with wash buffer. In this case, it could be PBST or TBST. Then please remember to use clean plastic forceps to transfer the membrane. Here you will see we'll do 5 times washing steps here. One time a quick wash, just a quick wash to rinse off to wash off the primary antibody. Next, a proper wash with the wash buffer for 15 minutes. Then 3 times wash with the wash buffer, each with 5 minutes. So you can see there are certain formulation of how many volumes that you should put in the use in each of these washing steps. So we'll say that this is a minimal requirement of the volume that you should use. For example, a standard 7 times 9 cm membrane will require almost 50 ml wash buffer for this 15 minutes wash, and around 20 ml of wash buffer for these 5 minutes washes. These steps is to make sure that the membranes are rinsed uniformly to remove the majority of the excessive primary antibody incubation solution. However, the gentle agitation must be done gently, softly, to prevent any scratches or damage to the surface of the membrane. Any scratches or damage to the blot surface of this membrane will create an active site or a lot of active sites that can absorb the secondary antibodies and gives you artifacts or high background. Okay, after primary blocking, primary antibodies wash, now we are at the stage we should do secondary antibody incubation. Same thing, prepare the secondary antibody mix with the blocking buffer. Gently mix it by inverting the tube. Do not vortex it. And same as primary antibody, we should optimize the dilution empirically. The recommended secondary antibody dilution for HRP secondary antibodies are either 1 in 5,000 to 1 in 20,000. A good initial dilution is 1 in 10,000. Same as primary antibody that we talked about just now. If you're using film detection for your Chemi-Westerns, use 5 times less secondary antibody than what you use in CCD imaging. A good starting point will be 1 in 50,000 dilutions. After you prepare the secondary antibody mixed with the blocking buffer, put it in the clean tray, transfer the membrane to this tray where it should be incubated for 1 hour at room temperature at gentle agitation. Right, after secondary antibody incubation, it's time to wash off the excessive secondary antibody. Same thing like how we wash off in primary antibody. Transfer the blot to a clean tray just for a quick wash with the wash buffer. Then we do 3 washes with the 5 wash buffer, each with 5 minutes. Right, here we proceed into imaging step, the final step before imaging, which is incubate with the ECL substrate. If the ECL substrate used in this step is azure radiant substrate to enhance the chemiluminescence signals. Firstly, mix the substrate to obtain sufficient volume for at least 0.1 ml per centimeter squares of the blot. This is a minimal working amount. Okay, so for example, if you're having 7 times 9 cm blot, this minimal volume is 7 times 9 times 0.1, which will give you 6.3 ml. If you are using minimal volume, the incubation can be done without agitation, like this. You just need to make sure that the membrane surface is leveled, so the reagent can be held on top of the membrane by surface tension. Here you can see you have the tray at the bottom, you have the blot where the proteins are facing up, and on top you put your radiance mix or your ECL mix and hold it on. Those will be hold on with a surface tension without leaking out of the blot. Keep it for 2 minutes. Same things if you are using more than not using the minimal working amount. This is for minimal volume. If you're using a lot more volumes, you can put it in the tray and then incubate for 2 minutes with gentle agitation. Right, allow the substrate to react with the blot for 2 minutes. Okay, one another thing is why we recommend you to use plastic forceps is and not metal, because metal forceps generally damage the block surface and creating the new absorption site. Like I said, that will give you the artifacts and high background. And also, the traces of metal on the membrane may act as a catalyst for non-enzymatic substrate oxidation, which will also give you very high background later on. All right. So finally, we should do the imaging step. Drain off the excessive substrate reagent by the capillary action. You can do it by touching a Kim wipe or other absorbance material and to the edge of the blot and remove the excessive substrate. While the blot is damp, it has to be wet all the time. It should not be dry. Cover it with transparent plastic wrap, or you can insert it inside a folder. Just keep it moist at all time. Place the blot in a CCD imager or expose the blot to a film. Here, these are the images that you should see if you are using CCD imager. For CCD imager, in terms of other imaging system, we allows uh, you can have the merge true color imaging here together with your Chemi blot. It's easier for you to recognize the molecular weight. And this is an image of a Chemi Western detected on X-ray film. Right. So here, this is the complete walkthrough of a detailed protocol of these chemiluminescence Westerns. Next, we move on to a troubleshooting tips. Right. If you see this type of protein, I'll say image, when you're trying to probe and strip and reprobe the membrane for a second protein of interest detection, you still see signals from the proteins that you detected firstly. That means that your membrane is not fully stripped. How you should do is ensure that you have enough stripping buffer volume and also stripping time. Thus, you will get distinctly two proper chemi images of your Westerns, detecting individual proteins at the same time. Right. These are the two images. If you detect the protein, not washing off the easier substrate before stripping, or you use the wrong buffer, or you use the wash buffer to wash off the easier substrate before stripping. This is what we say as insufficient or incorrect wash prior stripping. In order to do the stripping and reprobe process, firstly, we should rinse the blot with purified water for at least 5 minutes before the strip and reprobe process. And do not use wash buffer that containing twin to wash off the ECL substrate. If you're doing the strip washing and strip and reprobe process nicely, then you should get a clean and nice images after for the second images you take. Right. This is a chemi blot showing you the bubbles in the protein lanes. This means that you have all what we call as a white dot. These are the bubbles. To this means that the transfer sandwich is not either tight or you have bubbles in between that did not remove properly. So how you can do that is to remove these bubbles is press and smoothen the filter paper after covering the membrane to remove the bubble. There are certain I'll say there are certain practice on how to remove it. You shouldn't press it too hard, right? You should just roll it off using the specific roller. Okay, just remove the bubble. And also, please ensure that you have enough transfer buffer in the cartridge. Right. This is the image that you will see if your transfer is not having enough or uneven pressure. Thus, you will see the proteins are going haywire. They are not from one direction, A to B, like vertically. They are not doing that. So how you should do is the transfer setup should be very tight, and also place the gel and membrane in the middle of the cassette. If you're doing wet transfer, if there are 3 slots, then put it in the middle. If you're doing semi-dry transfer and put you are only transferring one blot, for example, and put it in the middle as well. Don't put it at the edge of the transfer apparatus. Right. So this is the image you will see if you have poor quality antibody, where you see some spearing and unspecific bindings. Firstly, what you should do is screen the antibodies just to make sure once you receive the antibodies, do a dot blot test to make sure they are working properly. They are not having poor quality and non-specific bindings. But of course, there are other causes for this particular image as well. For example, non-specific bindings of the primary antibody, or you just putting up too much primary antibody during your primary antibody incubation, or insufficient blocking. Perhaps your dry skim milk content is not high enough to provide enough blocking, concentration wise and also dilution wise matters. Right. So if you optimize this, you should get a clean background and Chemi Western image with specific bands that shown on the image as well. Right. I think this is a final troubleshooting tips, which is high background, where we will always see if after you put on the ECL substrate. So firstly, what you can do is reduce the primary antibody concentration by increasing the dilution factor. This can be done also first once you get your primary antibody, do a dot blot test, testing different dilution factor, okay, at different strip to get the optimal image. And you can also try a different blocking buffer. Let's say BSA doesn't work, try on skim milk then, right? Then also try a shorter exposure time when you use either both CCD imager or extra film, just to to get a very good balance between background noise and also the signal, your band signals. And finally, you can also increase the washing time and frequencies to wash out the excessive primary or secondary antibodies. Right. This is also another thing that we often see in amateur user, Western blotting user, which they don't see or they couldn't having low signals. Firstly, please check that you have the correct primary antibody used that to detect your protein of interest and the proteins that exist over there. One of the way it can be done is either is your primary antibody issue, or you can include another lane containing a positive control that you confirm that the protein of interest is present. Second, check the secondary antibodies that recognizes your primary antibodies of choice. For example, if your primary is an anti-rabbit, is a rabbit antibody, then the secondary antibody should be goat anti-rabbit. Okay, right. Speckled background. This is the image and you'll see a lot of dots, the unknown speckled dots on your blot. What you can do is filter the secondary antibodies because it might contain debris. Um, then also filter your blocking or wash buffer. I would recommend you, if you have enough resources, try to prepare fresh and secondary antibody blocking and wash buffer every single time, just to ensure that this will not be causing you an issue later on, because at the end of the day, Western blotting takes a lot of time and your sample might be precious as well. Right. So if you have done all these things and you still see the speckled background, ensure that your lab environment is clean, and to minimize the dust and debris on other particles that may come in contact with your blot. It might be on your table, it might be on the air. Try to cover the tray during incubation or washing step to avoid any unnecessary contact with these particles. Use a non-powdered gloves, or if you're using standard latex glove, try to switch on to a different kind of gloves. We recommend powder-free nitrile gloves and polyethylene gloves that will significantly improve this speckle background caused by these powders or dust. Right, there goes for our Chemiluminescence Westerns. Next is Fluorescence Western blot protocol. This case, I'll be using quantitative two-color assay to explain fluorescence Western blot. On the right, you can see these are the simplified workflow of fluorescent Western. This the same as Chemi Western as well. The only difference is what are the primary antibody or secondary antibodies that you use. As you can see in this schematic diagram, you have the same thing. You have proteins that transfer to the membrane with blocking compounds, blocking of the unnecessary site, protein of interest, primary antibody that recognizes the epitope, secondary antibody conjugate that has dye, fluorophores that binds onto it. So the only difference is here, instead of HRP, now you have fluorophores, which will excite at a certain excitation wavelength. On the left, you have the list of materials you should use in fluorescent Western. Same thing, primary antibodies. We recommend a combination of mouse or in rabbit instead of mouse or rat to avoid cross-reactivity. And of course, you need to have your electrophoresis apparatus, electroblotting apparatus, and also, yeah, one important thing is you shouldn't use nitrocellulose in membrane in Fluorescence Western. You should use PVDM membrane, especially low fluorescence PVDM membrane to give you clean background later on. Blocking buffer, wash buffer. These are the two secondary antibody conjugate, rapid 650 and mouse 550. Methanol to activate the PVDM membrane. PBS or TBS to wash or for final rinse without a twin, without a detergent, which is quite important. Faucets with flat, smooth tips. Powder-free gloves. Incubation and washing tray with smooth interiors. Rotary or rocking platform. And finally, the background quenching sheet. Okay, now let us look into the quick protocol. I will not go into details because there's a lot of parts going on. In this session, we'll focus on chemiluminescence westerns. Right. In terms of quick protocol of fluorescent westerns, firstly, prepare the blot like we usually do, run the electrophoresis. Okay, then transfer using wet transfer. We highly recommend wet transfer in fluorescent western. Block the membrane for 10 minutes, incubate the primary antibody, wash with washing buffer 2 times quickly and 3 times with 5 minutes each. Secondary antibody incubation, washing step as well, and the final washing step with the PBS or TBS without any detergent. Then put a blot on the quenching sheet and drain out the excessive liquid, then you're good to go for capture image. This is the reason why I said, or Western fluorescent Western is taking shorter time than chemiluminescence because you do not need to strip and repop to detect the second antibodies, and you do not need to cut off your blot into a smaller strip and then try to assemble them nicely for the imaging. All this can be done on the same blot. You don't need cutting, you don't need strip and repop process, and we can just get the image once for all by the end of the Western blotting protocol. All right, so this is an image I taken from the application note which you can download from Azure System website. This is a 3-color Western blot with Azure Spectra antibodies. The digital images of this 3-color Western using 600 images. On the left, we have 1, 2, 5, 10, 20 microgram of HeLa cell light. And this is a merged image of the 3 channels where green is the tubulin, red is beta actin, and GAP-DH in blue channels. Okay, so we have talked about detailed walkthrough of chemiluminescence Western, a quick glance at the fluorescence Western as well. Now here is, if you want this session is for those who want to improve your Western blot quantitation power with total protein normalization. In this case, we propose a Zoo 300Q imaging system for you. Right, just before I move into that details, is for some of you who might not join in the previous section. Last session, we talked about the differences or 2 types of normalization methods we are using in Western blot right now. Firstly is housekeeping protein, which we have been routinely used for the past many years, where it relies on a single band in that particular sample as an indicator or as a control for that particular sample or the entire membrane. And this is highly dependent on the high expression and the consistency of the expression across the experiment, either time points, tissue samples, and cell type, and so on. But in the actual case, it's not like that. We sometimes will see that I have maybe some of the samples that having low, for example, GAP-DH expression, but it doesn't mean that I have low protein load. It doesn't mean like that. It just might be happen to be that particular housekeeping protein is not housekeeping enough for that particular samples. In that case, that's why scientific community and even journal editors then be promoting scientific community to use total protein stains, because you are staining in this method, you are collecting the bands of a signal intensity of the multiple bands stained within the samples. So the differences is here. You have more than one, a lot is actually more than that, more than 1, 5, 10, more than that. bands for you to collect the signal intensity and use the collective amount of value as the control. In versus housekeeping protein, you are rely on this one single band as your control. That's why total protein stains, it minimizes the error and variability and reducing the risk of having I'll say risk of having perhaps not so accurate quantitation in your control groups or so on and so forth, which might not be noticeable right now, but it will be very obvious in the future, especially if you try to replicate your result in, let's say, a different lab or a different setting. Thus, we propose Azure 300Q, which is a Azure 300 imaging system plus Q module for you if you want to preserve your optimized Western blotting, Chemi especially, Chemi Western blotting protocol, but you want to enjoy the total protein stains to add on the quantitation power to your Western blotting, which I kind of understand because we have optimized everything, but I just want to, have more control in that. Thus, 300Q is a good starting point for you for your consideration. This is a Chemi imaging system. As you can see, at 300 here, you can do the normal gel documentation, UV and Chemi, plus include then for 300Q, you have extra Q module, stands for this fluorescence green channel. This fluorescence green channel is good for you to include and to view the total protein stains. In this case, what we talk about is azure red and total stain Qs, these two total protein stain dyes. And for these two for to do a total protein normalization on your Western blotting later on. One of the things is these two dyes can also be viewed under UV channel and also safe dye channel respectively, but of course the signals are not optimum. The optimum signals for these should be fluorescence green channel. Just a quick brief about these two dyes. These are both post-transfer labeling with no chemical bond. That means the process is reversible. You can just destain it at any time on your plot. And total stain Q is best for cell lysate, ranging from 1 to 50 microgram per lane, protein amount. Azurate is good or best for biological fluids with low protein concentration, ranging from 0.1 to 1 microgram per lane. So this is what you will see if you're trying to detect a total protein stain in the Chemi blot. You can pick the Chemiluminescence module, detecting just the CCD camera, and then picking one here, I pick Azure Red. And you can see on the right, this is the user interface. Chemi, of course, this is a Chemiluminescence light. There's no excitation and filter there. It's just a CCD camera. And you have Azure Red, which excite and absorb at green fluorescent channel. Thus 524 nanometer wavelength and 572 nanometer wavelength of excitation. Okay, so this is what you will get by the end of the day if you are staining your Chemi blots with total protein stains. You will get these fluorescence green total protein bands plus your Chemi signals together. And this is the user interface on the Azure 300Q, okay, that indicated by the colors of the bands. Thus, you have it. Thus, this is good if you want to do total protein normalization in your Chemi Western blot. Right. Let's recap what we have talked about today. Firstly, we go through or walk through the detailed protocol of Chemiluminescence Western blot and a few troubleshooting tips for Chemi Westerns, which are quite a normal practice, but it will be very useful if you are new to Chemi Westerns. A quick protocol on Fluorescence Westerns, which I didn't go in details because there are a lot more to talk about as compared to Chemi Westerns. But this is a trend right now for Fluorescence Western because it gives you definitely more quantitation power. And finally, if you just want to enjoy the quantitation power of total protein normalization, here we propose an Azure imaging system 300Q for you with additional fluorescence gray channel allows you to do total protein normalization on your Chemi blot. Yeah. If you're looking for Western blot reagents and imaging system for your lab or setting up a new lab, we Azuba system have a range of gel system, reagent, and also imaging system for your Western blotting needs. This is the Azure imaging system that I mentioned just now. We have Azure 200 to 600 models, all of them, except for 600, of course. Each of them can be upgradable on field without returning it to to our manufacturer site. If you're new to Western blotting and also you want to know all this information, please download the guidebook, Western blotting guidebook from the site, from the link shown here. It contains 100 pages of Western blotting guides covering both chemiluminescence and fluorescence westerns, tips, including tips, application notes, and references and consideration of each of every single steps. Thank you.